Terus sambung nih, roll.
Hello everyone. My voice is audible to everyone. Would you please let me know?
Yes, okay. It is, I think.
Okay, okay,
Yes,
okay.
sir, audible.
Okay, that's great. Thank you. Hello everyone. I'm very grateful to you, all of you, for joining this session, for joining the webinar on nutrigenetics and personalized nutrition. As you all know, the nutrigenetics and nutrigenomics has become an important topic in this clinical, in both in the clinical research and the clinical topic to treat and to prevent any illness or treatment and to promote and support health. So to talk on this topic, we have invited our esteemed guest, Dr. Vimal Karani. And before introducing Dr. Vimal, I would like to request Dr. Umar Bacha for a welcome note. Dr. Umar is an associate professor and acting chairperson at Department of Nutrition Science, School of Health Sciences, University of Management and Technology, Lahore. Over to you, sir.
Auzubillahi minash shaitanir rajim. Bismillahirrahmanirrahim. Hello and assalamu alaikum to everyone. Thank you, Mr. Fahim. This is Umar, and on behalf of School of Health Sciences, I warmly welcome you all for this webinar on nutrigenetics and personalized nutrition. Our today guest speaker is a distinguished professor in nutrigenetics and nutrigenomics, and he is presently working at the University of Reading. And we are thankful to you for sparing time. And you are welcome, Dr. Vimal Karani. Uh, Dr. Vimal obtained his postdoctoral training from Cambridge, UK, and from University of College London, UK. He published his work in renowned journals such as Nature Genetics, Lancet, PLOS Medicine, and PLOS Genetics, to name a few. We believe our participant will gain in-depth knowledge on personalized nutrition and nutrigenetics. I'm confident you will enjoy his experience and talks today. Thank you very much, Fahim. Over to you.
Okay, thank you very much, Sir Umar. Uh, Now I would like to call Dr. Vimal Karani, as Dr. Umar has already introduced Dr. Vimal in a very amazing way. In addition, I will add some few points. Like Dr. Vimal, actually, Dr. Vimal also has global media attention in the field of nutrition, and has helped numerous students and researchers due to his immense experience in the in the nutrigenetics research. Uh, so, so before delaying further, I will request Dr. Vimal Karani. So please so let's start. Thank you very much, Dr. Vimal, for joining us. I am turning over to you.
Thanks, Fahim, for a great introduction, and thank you so much for inviting me to this event today. So without any further delay, let me share the slides. Can you see the slides?
Yes, it's visible.
Yes, please.
Okay, let me. Okay, a very good afternoon to everyone, and hope you're enjoying the day. And thank you once again for having me today to give you a talk on nutrigenetics and personalized nutrition. So today I'm going to give you an overview about what is nutrigenetics and what is nutrigenomics, and how the findings from nutrigenetics can be used for implementing personalized nutrition. And for those who do not have a background in genetics, so let me start with some basic aspects of genetics and how the information from genetics can be used for merging with the nutritional science and leading to the field of nutrigenetics and nutrigenomics. So starting with some basics of genetics. So we know that every eukaryotic cell has got a nucleus, and inside the nucleus, you have the genetic material, and that genetic material is nothing but the DNA. So the DNA is made up of the nucleotides. So nucleotides are nothing but the nitrogenous base, which is attached to a sugar molecule, which is what we call as a deoxyribose, and which is attached to a phosphate molecule. So the base, the sugar, and the phosphate together constitute a nucleotide. And the human DNA is made up of 4 different nucleotides, namely A, C, T, and G, adenine, guanine, cytosine, and thymine. So the entire human DNA, if you look at the human DNA, there are about 3 billion nucleotides that have been identified by the Human Genome Project. And within this human DNA, we have several genes. There are about like 40,000 genes which have been discovered by the Human Genome Project. And what do these genes do? The genes code for the proteins, because these proteins could be any kind of a protein. It could be an enzyme, it could be a secretory protein, or it could be a transport protein, or it could be a membrane protein. So all these proteins, eventually they carry out various metabolic activities. So which clearly shows that any defect, any variation in the human DNA, any variation in the gene can have an impact on the quality and the quantity of the protein that is being synthesized. So if the proteins are defective, then the metabolic activities cannot be carried out. And if the metabolic functions are not being carried out properly, then that can lead to the development of diseases such as obesity and diabetes. So defects in the gene will have an impact on the protein that is secreted, which in turn can inhibit the metabolic functions and eventually lead to obesity and diabetes. So this is the basis on which we do all these nutrigenetics and nutrigenomics research. So looking at the genetic variation. So what is a genetic variation? So let's say this individual has got this particular DNA sequence, and you know that the DNA is double-stranded. And let's say this population has got these 5 individuals. And you can see that 99.9% of the human DNA is the same in all the individuals, except for 0.1% of the human DNA, which carries these genetic variations. And this variation, this 0.1% of the variation, is what is making you as a unique person. Because my DNA is very different from that of your DNA. I look different from you, and you look different from the person sitting right next to you. So we all look different in different ways. The reason is the difference in the DNA because of these genetic variations, where these genetic variations might be present in some people and it is not present in other people. So this 0.1% of the variation, if you look at it, you can see that some people carry T and some people carry A. So there's a change from one nucleotide to another nucleotide. There's a change from T to A. And this change from the one nucleotide to another nucleotide is what you call as a single nucleotide polymorphism, or in short, SNP. So this SNP is main reason why we all look different from each other. So if you take the human DNA, there are about 3 to 4 million single nucleotide polymorphism. So depending upon whether you carry T or A, the T is a normal variation, whereas the A is basically the genetic variation. So depending upon whether you carry 1A or 2A or none of the A nucleotides, you can classify individuals into 3 different categories. That is, if you do not carry any copies of the genetic variation, you're classified as a 0 copy individual. You don't carry any copies of the genetic variation. And if you carry 1 copy of the A and 1 copy without the genetic variation, then you're classified as a 1 copy individual. And if you carry 2 copies of the genetic variation and you don't carry any copies without the genetic variation, then you're classified as 2 copy individual. So 0 is where you don't carry any copies of the genetic variation, and 1 is where you carry just 1 copy of the genetic variation, and 2 is where you carry 2 copies of the genetic variation. So in general, people who carry 2 copies of the genetic variation, they have increased genetic risk of developing a particular disease. So here I'm talking about 1 genetic variation. Remember, our human DNA has got 3 to 4 million single nucleotide polymorphism. So which means that if you're going to carry 2 copies in all the genetic variation, then which means that you are accumulating your genetic risk. So what is this genetic risk? So let's have a look at what do you mean by this genetic risk. Say, for example, if you carry 0 copy, it means that low genetic risk, which means that you have decreased risk of developing the disease. So just because you carry 0 copy, it doesn't mean that you're not going to develop the disease. You still have a risk, but which is lower. And if you carry 1 copy, it means a moderate genetic risk, which means that you have moderate risk of developing the disease. And 2 copies, which means that you have a very high genetic risk, which means that you have increased risk of developing the disease. Just because you carry 2 copies, it doesn't mean that you're destined to become diseased. You can still overcome this genetic risk by modifying your lifestyle. How do you modify your lifestyle? That is doing lots of physical activity, consuming a healthy diet, which can overcome the genetic risk of a particular disease, such as obesity or diabetes. So this is the underlying basis on which you do nutrigenetics and nutrigenomics studies. So with this background, let's move into what is nutrigenetics and what is nutrigenomics. So talking about when nutrition meets genetics. So when I say nutrition, I'm talking about the traditional nutritional science. The traditional nutritional science is nothing but where we are all following the dietary recommendations which have been proposed by the WHO. We have all been following these dietary recommendations, that is consuming a balanced diet of carbs, proteins, and fats, and consuming 5 fruits a day. So all these kinds of dietary recommendations, which are universal dietary recommendations, are available, and we have all been following that for the last few decades. But still, there is increased prevalence of diseases such as obesity and diabetes. Why the prevalence is increasing when we are following the dietary recommendation? The main reason over here is the dietary recommendation, the universal dietary patterns might not be applicable for everyone. It might work for some people, but it might not work for other people. And the main reason is your genetics. The Human Genome Project has identified the human DNA sequence, and it has shown that we have 3 billion nucleotides in the human DNA. And within these nucleotides, you have 40,000 genes. And these genes are coding for different proteins. And these proteins are doing various activities in the body. So eventually, the genes are the ones which are regulating our physiological system. So we have to consume a diet which is suitable for our DNA. So when trying to merge the traditional nutritional science with that of the modern genetic world, the product is nothing but the nutrigenetics and nutrigenomics. The field of nutrigenetics is different from that of the field of nutrigenomics. But why do we do nutrigenetics and nutrigenomics research? The main reason is to implement personalized diets. That is like implementing diets which are suitable for an individual according to that individual's genetic makeup. If I carry 2 copies, what is the risk of developing a particular disease? And what is the kind of diet I should consume to overcome the genetic risk? So these are the questions that which are being answered as part of nutrigenetics and nutrigenomics research. So now let me move into what is nutrigenetics and what is nutrigenomics. So nutrigenetics refers to the study of the impact of DNA sequence variations on chronic disease outcomes in response to a particular diet. That is, for example, if I'm consuming a high-fat diet and I carry 2 copies, what is the risk of developing obesity? In order to answer this question, I need to see the impact of the high-fat diet and the 2 copies, that is high genetic risk, and I need to look at the interactions between the genetic risk and the high-fat diet and how they are interacting and contributing to the development of obesity. So this gene-diet interaction is what you call as nutrigenetics research. Whereas nutrigenomics, as the name implicates, omics refers to the expression, genomics refers to the gene expression. The word omics represents expression, and genomics is a gene expression, and nutrigenomics refers to the impact of the nutrient on gene expression. So what is gene expression? That is how the DNA is converted to RNA and how the RNA is converted to proteins. So you're looking at how the particular food that you're consuming, the food has got the nutrients, how the nutrients are entering inside our body, and how it's interacting with the DNA, and how it is regulating the expression of the DNA. That is DNA to RNA and RNA to proteins. So this study is what you call as nutrigenomics. And the findings from nutrigenetics and nutrigenomics are essential for the implementation of the personalized nutrition. What is personalized nutrition? That is, you're developing an optimum diet for an individual according to that individual's genetic makeup. Say, for example, if I carry 0 copy or 1 copy or 2 copy, what is the kind of diet I should consume? So that for understanding that dietary pattern, you need to do the nutrigenetics and nutrigenomics research. So nutrigenetics is nothing but the interactions between the genetic factors and the dietary factors, or in simple words, it's basically exploring the gene-diet interactions. Now, looking at the nutrigenomics. So what is nutrigenomics? Which is nothing but the study of the role of the nutrients in gene expression. That is, as I said before, the food that you are consuming has got the nutrients, and these nutrients need to be absorbed by the cells. Because that's the reason why we are all consuming food, because we need the nutrients and we need the energy from these nutrients. So these nutrients could be macronutrients or micronutrients. So these nutrients need to be absorbed by the cells. So the nutrient, which could be a macronutrient or micronutrients, I mean, it could be vitamins or minerals or amino acids or flavonoids, any kind of a nutrient. So these nutrients, basically, they go and bind to the cell and enter the cell membrane, enter through the cell membrane, reaches the cytoplasm. Once the nutrient reaches the cytoplasm, in the cytoplasm, you have certain molecules which are waiting for the nutrient to enter. And these molecules are nothing but the transcription factors. There are 2 important functions of the transcription factor. One is to bind to the nutrient. The transcription factors will have to bind to the nutrient. That is the first important function. And the second important function is to take the nutrient to the nucleus where you have the DNA. The transcription factors, along with the nutrient, they go and bind to the DNA. They nourish the DNA and give the energy for the DNA to get converted to RNA, to proteins, and proteins to metabolites. So in response to a particular nutrient, there are certain RNA molecules which are produced, and that study is called as transcriptomics. So omics means expression, transcriptomics is the mRNA transcript expression. And proteomics is protein expression, how the protein expression is taking place in response to a particular nutrient. And metabolomics is where you're looking at the metabolite expression in response to a particular nutrient. So all these omics approaches, they come under one big umbrella, which is a Nutrigenomics. But why do we have to do all these Nutrigenomics or proteomics, transcriptomics, metabolomics research? The main reason is if you have an abnormality in your gene expression profile, that abnormality, if you're able to detect at an early stage, you are giving yourself an opportunity to modify your diet, because your diet is the one which is determining your gene expression. So if you're consuming an unhealthy diet, there will be an abnormality in your gene expression. If you're consuming a healthy diet, then the abnormality can be rectified. So you're giving an opportunity for yourself to rectify that abnormality by changing your dietary pattern. So these differences in the gene expression, they serve as molecular biomarkers. So understanding these biomarkers will help us to prevent the disease. How do you prevent it? By implementing the personalized nutrition. That is, you need to consume a diet which is suitable for your gene expression. So that area of research is what you call as nutrigenomics. That is, trying to understand how particular nutrient can influence the gene expression. Whereas nutrigenetics is a completely different area of research, where you're looking at the gene-diet interactions. Whereas nutrigenomics is something to do with the gene expression work. So with this background, let's move on to the Nutrigenetic Studies. So today, with regards to my talk, I'm going to focus mainly on Nutrigenetic Studies. So what are the Nutrigenetic Studies that have been carried out in developed countries? So let's have a look at the status of nutrigenetics in Western part of the world. So it all started off with the discovery of the obesity gene. So this gene was discovered exactly 14 years ago by two independent genetic studies, and these two studies identified this gene at the same time. And once this gene was discovered, people strongly believe that if they carry two copies of this obesity gene variation, they felt that they are destined to become obese. So to clear this notion from the minds of public, the epidemiologist decided to test whether doing more levels of physical activity or consuming a healthy diet can that overcome the genetic risk of obesity. So that was the time everyone started to explore gene physical activity interactions and gene diet interactions. So now let's have a look at the hypothesis behind this gene lifestyle interaction. So we all carry different level of genetic risk, but the genetic risk could be a low risk or a moderate genetic risk or a very high genetic risk. That is, either you could be a 0 copy, or it could be a 1 copy, or you could be a 2 copy individual. But eventually they are all determining the development of the disease. So the question is whether changing your lifestyle, that is doing lots of physical activity and consuming a healthy diet, can that overcome the genetic risk of developing a particular disease? So that is what we want to answer now. So let me give you an example of a gene physical activity interaction. So I'm going to give you an example of my study where I have identified how doing more levels of physical activity can overcome the genetic risk of obesity. So this study was done in 20,000 individuals from the UK population. So where I stratified these 20,000 individuals into 4 different levels of physical activity, that is active, moderately inactive, and inactive. On the y-axis, you find the BMI, and the 3 color bars, the white, yellow, and green that you see, they're basically 0 copy, 1 copy, and 2 copy. So my first question is whether 2 copy people are having higher levels of BMI. And I can see that in all the levels of physical activity, I can see that the 2 copy individual, the green bar, is significantly higher than the 1 copy and the 0 copy individual, suggesting that if you carry 2 copies, then you have a higher risk of gaining higher BMI. But you can see that the effect is highly pronounced among the inactive individuals. Because people who are inactive, that is leading a sedentary lifestyle, not doing any exercise at all, you can see that their effect is 0.44. So what do you mean by this effect 0.44? That is, for every copy of the genetic variation you carry, that is, if you're 1 copy individual, your effect is 0.44 kilogram per meter square increase in BMI. That is, if you carry 1 copy in your DNA, then you're going to gain 0.44 kilogram per meter square increase in BMI. But if you carry 2 copies, which means that the effect is going to be doubled, that is 0.88 kilogram per meter square increase in BMI. So if you lead a physically inactive lifestyle, you're going to increase the risk of obesity. But whereas if you do some amounts of exercise, you can see that the effect is 0.2, which is half of what you see for the inactive individual.
वो मेरा सोफा है ना, आओ मेरे वाले सोफे पे बैठेंगे, तुम्हें कुछ कहे तो सही.
So individuals who lead a physically active lifestyle, their effect is 0.2, which is half of what you see for the physically inactive individual. So this clearly shows that if you lead a physically active lifestyle, you can overcome the genetic risk. But if you lead a sedentary lifestyle, you're going to increase the genetic risk. So this study clearly identified the fact that physical activity levels can overcome the genetic risk of obesity. Now let's have a look at the gene-diet interaction, how consuming a healthy diet can overcome the genetic risk of obesity. So this study was done in 11,000 individuals from the European population. So this study was done in like 5 different European countries, and we collected data from 5 different countries, and we carried out a meta-analysis. And these 11,000 individuals were followed up for 3.5 years. So which was basically a longitudinal study. So here we found an interaction with the protein consumption. So here in this graph on the x-axis, you find the protein intake in percentage. On the y-axis, you find the weight change per year. The 3 lines that you see, basically individuals carrying 2 copies, 1 copy, and 0 copy. And you can see that as protein consumption increases, as individuals increase their protein consumption, the annual weight change significantly increases for people who carry 2 copies. That is, you're carrying 2 copies and you're consuming high amounts of protein-rich foods, then your risk of obesity increases. Whereas for 1 copy people and the 0 copy people, the difference is not very significant. So this clearly shows that lowering the total protein intake, that is less than 17% of the total energy intake. If your protein consumption is less than 17% of the total energy intake, that will be an effective way to prevent weight gain in those who are carrying 2 copies of the genetic variation. So this is how you translate the findings from Nutrigenetics to personalized nutrition. That is, if I want to provide a personalized diet to people who carry 2 copies, then the best personalization of the diet for this particular population would be to advise them to consume a protein, less protein diet, that is a low protein diet, that is less than 17% of the total energy intake. I'm not saying that a proteinaceous diet is not healthy, but what I'm trying to say is anything in excess can have a negative impact on health outcomes, especially when you go to the European part of the world. The consumption of the animal protein is very high, and increased consumption of animal protein, we know that it has been clearly shown to be associated with increased risk of diabetes and cardiovascular disease. And the other example is from the same study from 11,000 people, but here we looked at the Mediterranean diet. So the Mediterranean foods are quite rich in omega-3 fatty acids, fruits, vegetables, and olive oil. And we know that consuming higher amounts of Mediterranean food is good for our health. And now we have been able to show that consuming the Mediterranean foods can overcome the genetic risk as well. So in this graph on the x-axis, you find the Mediterranean diet score, which means that higher the score, higher the consumption of the Mediterranean foods. On the y-axis, you find the weight change per year. The 3 lines that you see is basically 0 copy, 1 copy, and the 2 copies. And you can see that individuals... So individuals carrying 2 copies, you know that people who carry 2 copies, they have higher genetic risk of gaining more weight. But here, these 2 copy individuals, by consuming higher amounts of Mediterranean foods, they were able to reduce their weight gain. So even though you are 2 copies, if you're able to consume a healthy diet, you can still overcome the genetic risk of obesity. So for this European population, those who are carrying 2 copies, the best personalized diet would be increased consumption of the Mediterranean foods, which can overcome the genetic risk of obesity. So the take-home message from this study is basically the lifestyle factors and the genetic factors do not work independent of each other. Rather, they work together, they interact together, contributing to the development of obesity. So just because you carry 2 copies, it doesn't mean that you're destined to become obese. You can still overcome the genetic risk by consuming a healthy diet and doing good levels of exercise. Now let's have a look at the status of nutrigenetics in developing countries. Because if you look at different low or middle income countries, like whether you take South Asia or South Africa or South America or Southeast Asia or Western Asian countries. So in all these countries, the dietary patterns are so different. The genetic makeup is different. The geographical conditions are different. The environmental exposure, everything is different. And the levels of physical activity varies from one population to another population. So given all these differences, the nutrigenetics is the only answer to find a preventive solution to prevent obesity and various other cardiometabolic diseases. So I started this collaboration called as a genuine collaboration. Genuine stands for the gene-nutrient interaction. It's been 7 years since I started this collaborative effort. The main objective is to implement nutrigenetic studies in contrasting ethnic groups. These are the countries in which nutrigenetic studies have been implemented and some of the research is going on. And these are some of the publications from the genuine collaboration. These are the different funding organizations that have supported my nutrigenetic activities in various countries. So the main objective of this genuine collaboration is to look at nutrigenetic studies in each and every ethnic group. But why do I have to look at each and every country? Because of the differences in the genetic makeup. Because every ethnic group has got a unique genetic makeup. Because if you look at an African, you can easily identify that that person is from Africa. And if you see someone from India or South Asia, you can easily identify the ethnicity of the person. And if you look at someone from China, you can easily identify that the person is Chinese. The way you're able to recognize an individual is purely based on their genetic makeup, which is giving their unique personality and the facial expression, the way you eat, the way you smile, the way you walk, all the personality and the behavioral activities are completely controlled by your genes. So these differences in the genetic makeup is one of the reasons why we are doing nutrigenetic studies in each and every population. And there's a unique but varying dietary pattern. I'm not sure whether you have noticed it. Like if you take any country, if you take even Pakistan, northern regions of the Pakistan, they have a diet which is different from that of the southern regions of Pakistan. But as a whole, as a country, the diet is unique. The same applies to the Indian population or Bangladeshi population or Sri Lankan, any part of the South Asia. It's the same situation. So given the differences, you definitely need to look at the gene-diet interactions. And the prevalence of diabetes is very, very high, especially in the South Asian population. And there are no studies on nutrigenetics. So without nutrigenetics, there's no future for personalized nutrition. So that was the main reason why we started off with this genuine collaboration. So I'm going to give you some examples of the first nutrigenetic studies that I've carried out in different parts of the world. So the first nutrigenetic study that was done in the Asian Indian population. So you know that if you look at any South Asian population, the main source of energy comes from the carbohydrate, because the main source of energy is coming from the rice, which is quite rich in carbs. So we found a very interesting interaction with the carbohydrate intake. So here in this graph, on the x-axis, you find the carbohydrate energy percentage. That is a low, medium, and high. Low is where people consuming carbohydrates less than 300 grams per day roughly, and high is where like individuals consuming carbohydrates more than 500 grams of carbohydrate per day. On the y-axis, you find the risk of developing obesity, and the red bar are those individuals who carry 2 copies. And you can see that individuals carrying high genetic risk and consuming high amounts of carbohydrate-rich foods, they had 2.46 times increased risk of obesity, which means that if there is an Indian person who is consuming a lot of carbs, that is more than 500 grams of carbs per day, and carrying a high genetic risk, then the risk of obesity will be 2.46 times. So you know that, especially this study was done in the South Indian population. In South India, the consumption of carbs is even more higher compared to the North Indian population, because they do consume significant amounts of carbs, especially as part of the breakfast, lunch, and dinner, whether it comes down to a spicy food or even if it's a dessert. So the lowering the intake of the carbohydrate less than 300 grams per day might be an effective approach to prevent weight gain in this Asian Indian population. We also carried out another study in the Sri Lankan population, which is another South Asian population. Again, we found an interaction with the carbohydrate intake. So here on the x-axis, you find the tertiles of carbohydrate intake, low, medium, and high. On the y-axis, you find the waist-hip ratio. So waist-hip ratio is an indicator of the central obesity. And here the white bar with dots is basically low genetic risk, and the red bar is a high genetic risk. And you can see that individuals consuming high amounts of carbohydrate-rich foods, that is exceeding 600 grams of carbohydrate per day and carrying a high genetic risk, they had significantly higher levels of waist-hip ratio compared to individuals who carry a low genetic risk. So this clearly highlights the fact that if you carry a high genetic risk and you're consuming high amounts of carbohydrate, then your risk of central obesity increases. But interestingly, if you look at the Sri Lankan diet, which is almost the same as the Indian population, but the way they cook and the way they eat is completely different. If you look at this is a traditional, typical breakfast of the Sri Lankan population, so which is highly rich in carbs. And this is another dessert which is being consumed regularly in Sri Lanka and which is quite rich in carbs and also in saturated fatty acid. And the third study was done in the Indonesian population, which is a Southeast Asian population. This study was done in a unique ethnic group, which is called as a Minangkabau community, which is the world's largest matrilineal community. And here we looked at the Southeast Asian women. And given that Indonesians consume significant amount of meat, there was increased consumption of the animal protein. And here we found an interaction with the protein intake. So on the x-axis, you find the tertiles of protein intake. On the y-axis, you find the waist circumference. The red bar is those individuals with high genetic risk, and the white bar with dots, low genetic risk. So if you focus on those individuals who are consuming low amounts of protein-rich foods, you can see that individuals who have a high genetic risk, the red bar, despite having high genetic risk, you can see that they have lower levels of waist circumference. So which means that even if you carry a high genetic risk, if you consume low amounts of protein, you can overcome the genetic risk of having higher waist circumference. Because as low protein diet, I'm not saying that you should reduce your protein consumption. The low protein diet, the mean intake of the low protein diet tertile was around 70 to 75 grams of protein per day, which is equivalent to the dietary recommendations of the protein intake for the Indonesian population. So which means that if this population, if this Indonesian population, if they stick on to the dietary recommendations for the protein intake, then they can overcome the genetic risk of central obesity. Because the individuals who are in the highest tertile, they were actually consuming more than 140 grams of animal protein per day, 140 grams of protein per day, which is double the amount of what is required for the body, so which can increase the genetic risk of central obesity. So this is basically a traditional food of this Minangkabau community, which is called as a beef rendang, which is being cooked regularly, which is quite rich in meat and which is rich in animal protein as well. And the next study was done in the West Asian population, that is the Turkish population. Again, the consumption of animal protein is very, very high because in fact, like we did a tertile analysis and we found that the highest tertile, they were consuming more than 150 grams of animal protein per day. So in this graph on the x-axis, you find the low, medium, and high, and the y-axis, you find the central obesity. The red bar are basically individuals with 2 copies, and you can see that individuals consuming high amounts of protein-rich foods, that is exceeding 150 grams of protein per day, they had 3.3 times increased risk of central obesity if they carry 2 copies. So which is again a replication of the finding that we were able to see in the Indonesian population. But you have to think of another way of how this is happening in different parts of Asia. Because if you look at the Indonesian population, the way they cook and the way they eat, the meat is completely different from that of the way that is being consumed in the Turkish population, because the Turkish population, the cooking style and the way they consume the meat is completely different. So we need to focus on these various aspects of cooking style and how much of nutrients is being present when these foods are being consumed. That would be a very interesting aspect that needs to be analyzed as well. So the last example that I'm going to give you is the first nutrigenetic study that we did in the West African population, that is the Ghanaian population. And Ghanaians are known for consuming high amounts of fat-rich foods. So in this graph on the x-axis, you find the total fat intake, which is in as a binary category, that is less than 47 grams of fat per day and greater than 47 grams of fat per day. And on the y-axis, you find the waist circumference, which is again an indicator of central obesity. And the black bar are those individuals with high genetic risk, and the white bar, low genetic risk. And you can see that individuals consuming more than 47 grams of fat per day and carrying a high genetic risk, they have significantly higher levels of waist circumference. So the best way to provide a personalized diet for this Ghanaian population would be to reduce the intake of fats, that is reducing the saturated fatty acids. So their consumption of fats is very, very high as part of their traditional dietary pattern. So reducing the consumption of the fat-rich foods might be an effective way to prevent weight gain in this population. So in summary, the genuine collaboration provides the first evidence of all these gene-diet interactions, and we were able to show that the genetic risk of obesity is being modified by the intake of carbs, fats, and protein. And these dietary patterns vary from one population to another population. So in terms of implementing a personalized diet for a particular country, you need to use the findings from only that population. If I want to implement a personalized diet for people from Pakistan, then I need to use the findings only from Pakistan. I cannot use the findings from UK and use that for implementing personalized diet for the UK population or for the Pakistani population. I have to use the findings only from the Pakistani population. So for which you need to carry out the nutrigenetic studies in this population, the Pakistani population, to identify the gene diet interactions and how they could be used for implementing personalized diets. So if you're interested in knowing more about what I'm doing as part of the genuine collaboration, feel free to go into this publication, so which was published last year, and which will give you a better understanding of like what's being exactly done as part of the genuine collaboration. So besides your DNA, because the food that you're consuming is interacting with your DNA, but it is also interacting with the gut microbiome. The food is going through the gut, and the gut has got different microorganisms. And these microorganisms in the gut, they weigh up to 2 kilograms. So which means that they not only comprises of the bacteria, but also include the viruses, phages, and yeast. There are various microorganisms. You need to have a good proportion of both good and the bad bacteria to carry out various activities. And it's very much important that you need to consume a diet which is providing the sufficient nourishment for your gut microbiome. Because the gut microbiome releases various molecules which are basically the short-chain fatty acids. So these short-chain fatty acids include the acetic acid, butyric acid, and propionic acid. And they maintain good levels of immunity in your body and maintaining your cognition levels, your mental health state, and also maintaining your physiological system. So the gut microbiome also is very crucial, and the gut microbiome that is present in my body is different from that of the microbiome that's present in your body. So which is also another unique component similar to the DNA. And the third important one is basically the metabolic profiling, because the foods that you're consuming, they're eventually getting metabolized and produced as metabolites. And the metabolites present in an individual is actually a unique profile. So it's not only DNA, it's not only the gut microbiome, but also the metabolic profile, which will determine the health of an individual. And how do you do this metabolic profiling? That is, you can get any kind of a sample. Whether you can do a human study or an animal study, you can get the blood sample, and from the blood, you can get the serum or plasma, you can get the urine sample or the fecal sample, the stool samples, and also the breast milk. So depending upon your question, you can carry out any kind of a study, and there are different metabolic profiling technologies that are available, such as NMR or mass spec or different kinds of chromatographic technologies which are available. But for example, if you're analyzing 1,000 samples and the data that you're going to get is massive, so you need to have kind of a sophisticated software to carry out all this data analysis and statistical analysis. And eventually, the ultimate aim is to identify the levels of the metabolites in each and every individual. For example, if I'm consuming a high-fat diet, I'm going to have certain metabolites which are produced in excess. If another person is consuming a very low-fat diet, and that person might have very low levels of a particular metabolite. So these differences in the metabolites will give you a better understanding of the molecular basis of health and disease. So based on these profiles, you can design the diet for an individual. So based on the gut microbiome, based on your metabolic profile, and also based on your genetic risk, you're designing a particular diet. And that diet is what you call as a personalized nutrition or the personalized diet. So personalized nutrition is nothing but the tailoring of the dietary advice to suit an individual based on that individual's genetic makeup. So when talking about translating the findings from nutrigenetics to personalized nutrition, it all starts with the diet. The food that you're consuming is going into three different compartments. The first compartment is basically the gene, the DNA, so which we have already seen as nutrigenetics or the gene-diet interaction. And the second compartment is basically the gut microbiome, the gut, where the diet interacts with the microorganism. It nourishes the microorganisms and helps you to produce all the good molecules which are required for maintaining a healthy physiological system. And that area of research is metagenomics. And the third area of research is basically the metabolomics, where you're looking at the metabolic profiling. So you need to look at all these three different compartments, because the food, as soon as you consume, it is interacting with all these three compartments in parallel. So when you're trying to implement personalized diet, you need to look at it as a three-way interaction, metagenome, hyperbolome, diet interaction. And understanding this interaction is very crucial for implementing personalized nutrition. So this is how you implement a personalized diet, taking into account of the data from nutrigenetics, nutrimetabolomics, and gut microbiome. So in terms of looking at the universal dietary patterns, so some people could be a keto diet follower, or some people could be vegetarian, some people could be consuming a lot of non-vegetarian foods, and some people might be gluten-free foods. These are not unique foods. These are all universal diet. But you need to understand that you should consume a diet which is suitable for your DNA or for your genetic makeup. So that is what is very much required to overcome the genetic risk. So how do you do that? And that the way by which you implement that personalized diet is making use of the findings from nutrigenetics and nutrigenomics. Because the universal dietary patterns, it is not applicable for everyone. One size fits all approach doesn't work anymore. We are all looking for a personalized approach now, and that is going to be the future of how nutrigenetics is revolutionizing the field of nutrition, right? So on this basis, my conclusion is basically the healthy lifestyle can overcome the genetic risk of diseases such as obesity. As mentioned before, the genetic factors and the lifestyle factors, they can interact and contribute to the development of the disease. But as long as you consume a healthy diet and doing high levels of physical activity, even if you have a high genetic risk, you can still overcome that genetic risk and you can prevent the development of obesity. So if you ask me a question, is it the diet which is important or is it the DNA that is important? Definitely DNA is very, very important, because you are born with a particular DNA sequence. You're born with 0 copy or 1 copy or 2 copy. From day one of your life until the last day of your life, you are living with your DNA. You cannot change 0 copy or 1 copy or 2 copies. What you can change is your diet during your lifetime. You can change your diet. You can change your lifestyle. The lifestyle factors are modifiable factors, whereas DNA is a non-modifiable factor. You cannot touch your DNA. So you have to change your diet. So diet plays a very important role in overcoming the genetic risk of chronic diseases such as obesity, diabetes, and cardiovascular disease. So on this note, I would like to thank all my genuine collaborators, and also thank you so much for inviting me to give this talk. And I hope you have all enjoyed this lecture. And if you have any question, please do drop me an email at b.karani@reading.ac.uk. And I'm available on Facebook, LinkedIn, Instagram, and Twitter as Verma Karani. So if you have any questions, please do not hesitate to drop me an email. But before I finish, I would like to share one important announcement about a workshop, which is an upcoming workshop on Nutrigenetics, which is going to be on the 29th of May. And it's a workshop on Nutrigenetics, personalizing nutrition based on genetics. It's a one-day workshop. It starts in the morning and it finishes in the evening. It's a one-day fully packed with talks from various areas of research, starting with some ABCs of genetics for nutritionists and dietitians. So starting right from the scratch about basics of genetics, and then introducing you all to nutrigenetics and nutrigenomic concepts. And also there's a session on sports nutrigenetics, and also trying to help you in understanding how to translate nutrigenetics findings to personalized and precision nutrition. And also we are inviting a speaker from a company, which is the Adams Genetics company, and which is a leading DNA-based diet and fitness testing company. And he is going to give you some genetic reports and giving you some hands-on experience of how to interpret the data and how to translate that to personalized nutrition. Of course, all the attendees will get a certificate which is being approved by the University of Reading, UK. And if you're interested, please do contact Tausif. I'll share the brochure with Fahim, and he'll be able to circulate the brochure to you all. So thank you so much. If you have any questions, just let me know.
Thank you very much, Dr. Rumal, for this very informative session. So if you have any question, so you can ask in the chat box, or you can simply raise your hand to ask your question.
Assalamu alaikum, sir.
Yes, please, Ruksair, you can ask.
Yes, I have a question. The question is that the lecture was very fantastic and very informative. My question is that, so we have learned this thing, the diet which came from, especially Mediterranean diet, the diet which came from fruit and vegetables. So I have a question, the diet which came from, because now our cultivation process and our fertilization as well as agriculture techniques are totally changed. We are using fertilizer as well as different pesticides, and there are a lot of chemical material on our foods. So my question is that the food which came from and that is cultivated and harvested with pesticides, so if we consume that feed, either this is the same impact on our nutrigenetics and the food which we can say that organic food which came from a very organic material, there were no chemical introduction on the food. So if we compare these two food, which is the best food, or either the food, the question is that I am consuming fruit and vegetable, but they are totally contaminated with chemicals. Either they have the same impact on my nutrigenetics, or I will get the same result.
Hey, thanks for raising that question, but like your question is completely a different area of research. So that has got nothing to do with nutrigenetics, because that area of research is something called as the gene chemical interaction. Because what I was talking about is the gene diet interaction. Your question is something about the chemicals which can have an impact on the health of the individual. And these chemicals like fertilizers or pesticides and insecticides, these chemicals can also induce obesity. And these chemicals have been named or termed as obesogens. Like the way you have carcinogens which can induce cancer, these obesogens can induce obesity in individuals. And how do they induce obesity is that these chemicals, they enter into your body, it enters into the cell, and these chemicals have got a unique property which can have a pretentious phenotype as similar to a hormone. So these hormones generally, they enter inside, they are being produced and they are being absorbed by the cells. So instead of these hormones, these chemicals, they take the shape of the hormone, and these cells will assume that these chemicals are hormones and start consuming these chemicals. So eventually what happens over a period of time, these chemicals will start ruling all the cells, and eventually a person will lose that ability to produce a normal hormone, and eventually these chemicals will result in the development of obesity through inflammatory process. So your question is more to do with how the chemicals are being interacted with the DNA. But like what I'm saying is about like how the diet. So the gene diet interactions can be influenced by these obesogens as well. So they can also have an impact in contributing to obesity, and that's what the study says. I can send you an article about the paper that I published regarding this gene chemical interactions and how they can result in the development of obesity and diabetes.
Thank you so much.
Thank you, sir. Another question, a few participants are asking that what are your future goals or future plans for nutrigenetics, for study on nutrigenetics in Pakistan?
Sorry, I didn't hear you. There's a lot of background noise. Can you repeat your question?
Okay. A few participants are asking, like, what are your future plans in Pakistan about the research in nutrigenetics in Pakistan?
Future plans for nutrigenetics. Well, at the moment, like I basically, sorry. Yeah, so I'm basically interested in implementing the nutrigenetic study in Pakistan. The main reason is the resource, which is not being available. We had applied for funding to do nutrigenetic study in Pakistan, but that was not approved for various reasons. And now we are collaborating with a couple of teams in Pakistan where we are planning to do that study. And because first of all, we need to do a pilot study to understand the genetic profiling, and also then looking at the dietary patterns. And of course, Pakistan has got like kind of a heterogeneous population. So it's very much important that you need to do the nutrigenetic studies in each and every parts of Pakistan, because because I've been to Pakistan a few times and I've seen that the dietary patterns are so different across different regions in Pakistan. So definitely we need to look at the dietary patterns and the genetic profile, and studies need to be conducted in different areas to get a better understanding of how personalized nutrition services could be implemented. But what I'm doing at the moment is bringing awareness on nutrigenetics, because that is very, very crucial. Because if you don't bring that awareness on nutrigenetics and personalized nutrition, as a nutritionist or a dietitian, you will not be able to convince the public that there is a field of nutrigenetics. So I'm trying to bring that awareness on nutrigenetics and personalized nutrition through conducting these kind of webinars and workshops to bring some understanding not only to the public but also to the academics and students and nutritionists and dietitians from various hospitals. So that's what I'm doing as part of the genuine collaboration at the moment.
Thank you so much, Dr. Mal. A few students are asking questions in chat box. Can you please just review that?
Yeah. Okay, how can we know that how many copies we have through DNA test? Well, the DNA testing is otherwise called as genotyping. So the genotyping is basically a protocol where you basically trying to see, where you basically can get the blood sample, or you can get the saliva sample, or you can get a buccal swab. So these samples can be used for extracting the cells. You get the cells, you break open the cell, you break open the nucleus, you get the DNA out. And looking at the entire DNA, you can look at different genes over there. And within these genes, you can look at different genetic variations, and also to identify whether the person carries 0 copy or 1 copy or 2 copy. So that protocol is called genotyping. And there are several genetic testing centers. I'm sure there must be some genetic testing centers in Pakistan as well. And if you give your blood sample, they'll be able to tell you whether you carry 0 copy or 1 copy or 2 copy for the obesity gene. And the next question is, if we move to personalized nutrition, then how will we know about the person's gene expression for different food and nutrients? Well, to get the gene expression profile, there are different technologies that are available. You can do an RT-PCR gene expression. So the same way You can get the blood sample, and from the blood sample, you can do all these gene expression profile, that is getting the DNA out, and DNA is being, it's kind of like a complicated methodology. So these aspects are being covered in the workshop, and how to do a gene expression profiling and how to do the genotyping. So all these are being covered in the workshop. If you're able to attend the workshop, that will be very helpful for you guys.
Okay. Laiba Suman, do you have any question?
And if we are looking, sorry, I'm just reading out another question.
Yes, yes, please.
So what about carbs percentage while we are having protein less than 17%? And there's a similar question again. How will cutting the proteins less than 17% helps to gain muscle mass? Well, remember one thing, the data that I was presenting is based on one genetic variation. So as I said in the beginning of my lecture, our human DNA has got about 4 to 5 million single nucleotide polymorphism. So you cannot provide a personalized diet based on just one genetic variation. So when you are trying to implement a personalized dietary regimen, you need to take into account of panel of different genetic variations. So for one SNP, it could be a protein consumption less than 17% of the total energy intake. For another person, for another genetic variation, it could be where the person might be zero copy, which means that the person has got very low risk. So where you can increase the protein and you can increase a little bit of the fat because the person could be underweight. So you need to take into various factors. That is not just the genetic makeup alone, because when you do a personalized nutrition, you're looking at the genetic makeup, you're looking at the metabolomic profile, you're looking at the gut microbiome profile, you're looking at the biochemical profile, you're looking at the marker profile, you're looking at the socioeconomic status of the person, and you're also looking at the ethnicity of the person. So you're looking at various factors. So not just based on one SNP. You're not providing a personalized diet just based on one SNP, but rather you're looking at several factors together. What is your belief about keto diet? I don't believe in any of these diets, paleo diet or keto diet, or any of these universal dietary patterns, because as I said before, they are all one size fits all approach kind of a diet. And I'm a person who believes strongly in the personalized dietary approach, because our DNA is unique and you should be consuming a diet which is suitable for your DNA. So there's another question. Does every patient need to get a genetic report before diet planning? Well, it depends on like whether you're providing a personalized dietary service. So I know like a lot of nutritionists and dietitians, you're not taking into account of the genetic makeup or genetic profile at all. So if you are planning to use a genetic profiling, then you should have data available for the Pakistani population. So if you don't have the data on the Pakistani population, which means that you cannot use a personalized nutrition service for the Pakistani population. So which is a clear indication of why you definitely need nutrigenetic studies in Pakistan. So is intermittent fasting has positive effect on gene expression? So see, the intermittent fasting or periodic fasting, and there are several studies that have looked at the religious fasting and their impact on various measures of obesity, diabetes, and cardiovascular disease. And studies have shown that intermittent fasting has positive impact on the gene expression, and also it requires for the adipogenesis, that is for the adipocyte differentiation, which can reduce the risk of obesity. But all these studies have been done mainly on animals. We do not have any solid evidence on humans. And that is why the findings relating to fasting with regards to the human studies, they are not reliable, because we still do not have a solid evidence for humans. We do have evidence from animal studies where we have shown that intermittent fasting has got positive impact on anti-aging mechanisms, reducing the inflammatory effects and improving the adipogenesis process, which are all good for a healthy individual. But all the studies have been done mainly in animals, not in humans. And does Nutrigenetics has an effect on blood glucose level? Definitely. New gene diet interactions have been reported on several factors relating to diabetes and cardiovascular disease. So blood glucose level, blood insulin level. I can send you articles relating to my work, which I've done in South Asian populations, and where we have looked at the gene diet interactions on various diabetes related outcomes. And there's a question, foods helpful for osteoarthritis and osteoporosis? Well, I'm not a nutritionist. I'm more of a genetic epidemiologist. So I'm not in a position to suggest foods that can be helpful or to prevent osteoarthritis, because you please do consult a nutritionist or a dietitian. Because, of course, as I said, the diet has to be unique for each and every individual. And I don't believe in any universal diets. So again, there's another question regarding the keto diet. My response is still the same. So I cannot suggest you any kind of a diet relating to any of the disease outcome. That is very difficult on my part, purely because of the fact that the one size fits all approach doesn't work, and it works mainly with a personalized approach.
Thank you very much, Dr. Verma Karani, for your precious time and for honoring us to learn from your experience, or from your research-based experience and from your clinical experience. Now, at the end, we have a group photo session. So everyone, please turn on your cameras for photo session. Okay, all right. Thank you very much, Dr. Rimal. No doubt, I believe this session will be helpful for both students and faculty to collaborate with the University of Reading and University of School of Health and University of Management and Technology in the future, inshallah. Thank you very much for your time.
Thank you. Thank you very much. Have a nice day.